Imagine, if you will, a bustling metropolis, a city teeming with life, each building humming with activity, every individual going about their daily tasks. But what powers this vibrant city? What makes the trains run, the lights glow, and the countless gears turn? It's energy, of course. And just like any complex system, our cells, the microscopic architects of our very being, also require a constant, readily available source of power. Adenosine Triphosphate, ATP, the undisputed, dynamic, and utterly essential energy currency of life. From muscle flexes to silent thoughts, every flicker of life is an ATP transaction. It's a cryptocurrency far more vital than Bitcoin. The dawn of discovery, peeling back the curtain. Our story begins not with a bang, but with the quiet curiosity of dedicated scientists, meticulously probing the secrets of life. The initial whispers of ATP's existence surfaced in the early 20th century. The year is 1929. Imagine a dedicated biochemist, a scientific detective, in his laboratory, much like a meticulous code auditor examining complex digital transactions. This was Karl Lohmann, a German biochemist. He was deeply engrossed in studying muscle extracts, trying to decipher the elusive compounds that fueled muscle contraction, the very process that allowed us to move, to breathe, to live. In his biochemical explorations, Lohmann isolated a novel organic phosphate compound from muscle tissue. He carefully characterized it and, in a landmark paper, described its chemical structure. This compound, he realized, contained adenine, ribose, and a string of 3 phosphate groups. He named it adenosine triphosphate, or ATP. It was a crucial first step, like discovering the purest vein of digital ore, a powerful, mysterious element in the cellular landscape. You could say Lohmann was like one of the early, unsung pioneers of cryptography, recognizing a fundamental building block before its true power was understood. However, Lohmann wasn't alone in this groundbreaking era. Simultaneously, across the Atlantic, in the United States, Cyrus Fisk and Yellow Brigada Subaru were also independently making similar discoveries, contributing to the burgeoning understanding of this vital molecule. Their work, published around the same time, solidified the initial identification of ATP. Think of them as other early developers or miners in the nascent field of cellular energy, all converging on the same critical discovery. Identifying ATP was one thing, but understanding its profound significance in the grand scheme of biological processes was another. This deeper insight came from the brilliant mind of Fritz Lipmann. The year is 1941, amidst the tumultuous backdrop of World War II. Imagine Lipmann, not just a scientist, but a visionary. Much like a Satoshi Nakamoto for the biological world, conceptualizing the underlying mechanism of the new currency. Lipmann, a German-American biochemist, was deeply immersed in studying the metabolic pathways of cells, particularly the breakdown of glucose. He observed that many enzymatic reactions within the cell were coupled to the synthesis and breakdown of ATP. He meticulously theorized that ATP wasn't just another molecule. It was the universal energy carrier, the very compound that captured and transferred energy from energy-releasing reactions, like cellular respiration, to energy-requiring processes, like muscle contraction, protein synthesis, and active transport. Lipmann's most pivotal contribution was his groundbreaking concept of the high-energy phosphate bond. He proposed that the bond between the second and third phosphate groups in ATP held a significant amount of chemical energy, and that the hydrolysis, breaking of these bonds, released this energy for cellular work. This was a revolutionary idea, providing the fundamental framework for understanding how ATP actually works as an energy currency. He famously illustrated this with the wavy line, tilde-p, to denote these high-energy bonds. He wasn't just finding a molecule, he was essentially writing the white paper for life's energy protocol, explaining the fundamental principles of how this cellular cryptocurrency operated. For his profound insights into the energy metabolism of living cells, particularly his elucidation of the high-energy phosphate bond and the central role of ATP, Fritz Lipmann was awarded the Nobel Prize in Physiology or Medicine in 1953, sharing it with Hans Adolf Krebs for his discovery of the citric acid cycle. It was a richly deserved recognition for a scientist who had illuminated one of life's most fundamental mechanisms. Our understanding of ATP didn't stop there. The ability to synthesize ATP in the lab and the precise mechanisms of its synthesis were further illuminated by other brilliant minds. In 1948, Alexander Todd achieved a landmark by first synthesizing ATP in the laboratory. This was a crucial step, allowing scientists to study and manipulate ATP outside of living cells. For his work on nucleotides and nucleotide coenzymes, including the synthesis of ATP, Alexander Todd was awarded the Nobel Prize in Chemistry in 1957, partly for this groundbreaking achievement. The intricacies of ATP synthesis at a molecular level were further unraveled by Peter Dennis Mitchell. He proposed the chemiosmotic mechanism, explaining how the energy from electron transport is used to create a proton gradient that drives ATP synthesis. Peter Dennis Mitchell was awarded the 1978 Nobel Prize in Chemistry for this revolutionary discovery. The final pieces of the puzzle regarding the enzymatic mechanism of ATP synthesis were elucidated by Paul D. Boyer and John E. Walker. Their work detailed how ATP synthase, the molecular machine responsible for ATP production, functions. Paul D. Boyer and John E. Walker jointly received one half of the 1997 Nobel Prize in Chemistry for their contributions. The other half of the prize was awarded to Jens C. Scow for his discovery of an ion-transporting enzyme, highlighting the importance of ion gradients in cellular processes. Our cells are incredibly efficient. ATP is not just made and then discarded. After it releases its energy, it becomes ADP, adenosine diphosphate, an inorganic phosphate pi. The good news? Our cells constantly reattach a phosphate group to ADP, turning it back into ATP, ready to be recharged and used again. It's an endless cycle of energy, making ATP the most sustainable energy source known. An average human adult turns over their own body weight in ATP every single day. Think about that for a second. Your body is constantly making and breaking down ATP, a staggering feat of molecular engineering. If you weigh 70 kilograms, you're producing and consuming 70 kilograms of ATP daily. It's a testament to the incredible metabolic activity within our bodies. This constant turnover highlights why ATP is so vital. It's always in demand, always being replenished. Ever seen a firefly glow on a warm summer night? That magical light, known as bioluminescence, is powered by ATP. In fireflies, ATP reacts with a molecule called luciferin and an enzyme called luciferase, producing light. It's a direct example of chemical energy being converted into light energy, all thanks to ATP. From firefly glows to human thoughts, ATP is the universal spark igniting life's most amazing phenomena. When an organism dies, oxygen supply to cells ceases, halting ATP production. Without ATP, the muscle proteins actin and myosin remain locked together, unable to detach. This causes the characteristic stiffening of muscles known as rigor mortis, which eventually subsides as cellular structures begin to break down. It's a stark reminder of ATP's crucial role in muscle function. Even in death, its absence tells a story. While we focused on human cells, it's a fantastic fun fact that plants also produce ATP. In photosynthesis, during the light-dependent reactions, light energy is captured and used to generate ATP and NADPH. This ATP then fuels the synthesis of glucose in the Calvin cycle. So, in a way, the energy from the sun is first converted into ATP in plants, before being stored in sugars. This means the very food we eat carries the legacy of ATP from its plant origins. From muscle flexes to silent thoughts, every flicker of life is an ATP transaction. Meet Karl Lohmann, the first to glimpse this cellular gold, like discovering the purest vein of digital ore. Fritz Lipmann, the visionary who deciphered ATP's high-energy secret, essentially writing the white paper for life's energy protocol. Glycolysis, your cell's quick cash ATM, perfect for those small, immediate energy bursts. The mitochondria, the grand central bank vault, where the vast majority of your cellular wealth, ATP, is minted. ATP synthase, the ultimate molecular turbine, converting a proton flow into pure energy, like a hydro-powered crypto miner. Did you know you recycle your own body weight in ATP every single day? That's commitment to sustainability. From firefly glows to human thoughts, ATP is the universal spark igniting life's most amazing phenomena.